From spark to heartbeat, how a baby forms, on how a baby forms, ready. It begins when sperm meets egg, fertilization. That single fused cell is a zygote. The zygote divides quickly, forming a ball of cells, racing down the fallopian tube. Around day 6 to 10, it implants in the uterus. That's the nest where development continues. Cells organize into layers and early organs. Brains begin as tiny maps. Hearts begin as tiny maps. Limbs begin as tiny maps. The fetal stage is growth and fine-tuning. From heartbeat to kicking. Then, months later, labor and birth. Biology in motion. One tiny start. An incredible 9-month journey. Want a deep dive on any step? Tell me which one. We'll use gestational weeks, the calendar most clinics follow. Around week 3, fertilization occurs. One cell begins the story. Cleavage starts within days. Blastocyst by day 5, implantation around days 6 to 10. Weeks 5 to 10, the embryonic period. Organs sketch in. Cells fold. Tubes form. And first chambers appear. By week 11, we enter the fetal period. Growth and refinement take over. Features sharpen, and systems coordinate. Around weeks 3 to 4, gastrulation maps the body plan. 3 layers, ectoderm, mesoderm, endoderm, set destinies. Ectoderm becomes skin and the nervous system. Mesoderm forms muscle, bone, and blood. Endoderm builds gut, liver, and lungs. This is a sensitive window. Neurulation begins. The neural tube folds and closes. These steps lay out brain and spinal cord, guided by precise signals. Patterns are steered by cues like SHH and BMP. Folate supports proper tube closure. Weeks 4 to 8, early organogenesis accelerates. Limb buds sprout and lengthen. The heart loops, partitions, and moves blood efficiently. Pharyngeal arches sculpt the face and neck. It's the most vulnerable phase to teratogens. Now the placenta takes center stage. Chorionic villi exchange oxygen and nutrients with mom. Waste products return through the placenta. Hormones like HCG and progesterone support the pregnancy. Mom's immune system adapts to allow tolerance. Fetal circulation uses clever shunts to route flow. The ductus venosus bypasses most liver flow. The foramen oval moves blood between the atria. The ductus arteriosus sends blood around the lungs. Before birth, oxygen comes via placenta, not by breathing. Growth is tracked early by crown rump length. A heartbeat is often visible on ultrasound around weeks 6 to 7. First flutters, quickening, commonly appear between weeks 16 and 20. Screening snapshot, ultrasounds check anatomy and growth on schedule. NIPT, a blood test, is often available from about week 10. Timing each step to development keeps results meaningful, and worry low. It's a coordinated journey, resilient and precise. Which deep dive next? The placenta, fetal circulation, or neurulation? Before conception, meiosis shuffles chromosomes, crossing over mixes alleles, so recombination makes each gamete, and each embryo, unique. Many embryos are aneuploid. Nature screens early. Clinical screening aims to reduce risk, not guarantee outcomes. After fertilization, rapid cleavages begin. Cells compact and align, and start talking via polarity and adhesion cues. YAP and TEAD help specify the trophectoderm, while the inner cell mass will form the embryo proper. To implant, integrins help the blastocyst latch, and the endometrium decidualizes in response. HCG signals back to sustain the uterine environment. Patterning then unfolds through nodal gradients, WNT gradients, and BMP gradients, mapping head to tail and left to right. Notch and FGF oscillations pace segmentation, so somites pinch off rhythmically along the axis. Around the early embryonic weeks, the neural plate folds and the tube closes. Folate supports proper closure. Neural crest cells migrate widely to build diverse tissues. Limb buds extend under the AER's FGF, while the ZPA's SHH sets thumb to pinky identity. In the placenta, villi trade gases and nutrients via transporters, and IgG crosses via FcRn for passive immunity. Hormones from placenta and fetus modulate the maternal system. In utero, the highest oxygen content is in the umbilical vein, and shunts route flow efficiently, bypassing the lungs. Early split timing determines chorionicity in identical twins, while dizygotic twins are siblings simply sharing a womb. Because organs are forming, the embryonic window is most vulnerable, so timing matters for exposures. As birth nears, fetal and placental signals rise, surfactant production increases, Prostaglandins shift the cervix and uterus, labor ramps toward first breaths, pressures flip, and the shunts close in sequence. Which mechanism should we deep dive next?